Hi, I am Jocelyn Makasare, Assistant Professor, Department of Botany, Parvatibai Chogle College of Arts and Science, Marga. Program Bachelor of Science, Subject Botany, 5th semester, Paper Code BOC 106, Paper Title Cell Biology and Plant Biochemistry. The title of the unit is Techniques in Cell Biology. The module name is Introduction to Electron Microscopy, module number 3. Here we are going to start with the Introduction to Electron Microscopy. The learning outcomes: the student will be able to state the principle of electron microscopy, describe the basic construction of an electron microscope, and list the applications of an electron microscope. Introduction. Electron microscope is one of the most powerful tools used in cell biology to study the ultrastructure of organisms, both in eukaryotes and in prokaryotes. An electron microscope uses a beam of electrons to illuminate the object and form an enlarged image. Here, the electrons are the constituents of an atom along with protons and neutrons. A beam of electrons facilitates a higher resolution due to shorter de Broglie wavelengths. That is why electrons are used as an illumination technique in electron microscopy. Now, the resolving power of an electron microscope is 2 million times as compared to 2000 times of a light microscope. The electrons are used instead of light to illuminate the specimen. This is the basic principle of an electron microscope. The whole apparatus is situated in a vacuum tube, and a heated cathode emits a stream of electrons called cathode rays. Now, these cathode rays can be deflected and directed to focus using magnetic fields or electrostatic fields. The wavelength of the electron beam can be manipulated by altering the accelerating voltage. Electrons have wave-like properties as stated by de Broglie. The wavelength of an electron radiation used is about 0.005 nanometers. It is 100,000 times shorter than that of visible light. The resolution achieved is 1000 times superior to that realized by a visible light. So, with your high resolution and a high resolving power, the magnification in an electron microscope is enormously increased. Now, these are the signals generated by the interaction of the primary beam of electrons with a sample in electron microscopy. We have the primary beam of electrons, and when it passes through the sample, you get the transmitted beam of electrons. Now, these transmitted beam of electrons are used in transmission electron microscopy. We also have diffracted electrons and loss electrons. Now, there are backscattered electrons, secondary electrons, Auger electrons, X-rays and photons. Now, the secondary electrons, backscattered, Auger, photons and X-rays are used in scanning electron microscopy. These are the details of the signals generated. Now, transmitted electrons, the electrons pass through the object depending on the thickness and density without suffering any energy loss, and these provide a two-dimensional image of the object. Your backscattered electrons collide with the atoms of the object or your sample and get backscattered. Backscattering is effective with increasing mass of the atom. It provides information on distribution of different elements in the object. Secondary electrons are emitted by the object as the primary electrons lose energy due to inelastic collisions, and they provide information on the morphology and topography of the specimen. Auger electrons are emitted by the relaxation of core ionized atoms. X-rays are emitted by the relaxation of core ionized atoms, and they provide information on the chemical composition of the sample. Diffracted electrons, the electrons are diffracted by particles in the object, and they provide dark field images and crystallographic information about the specimen or your object. Loss electrons. Electrons emitted as the primary beam of electrons lose energy due to vibrations in the object. Photons. Emission of photons in the UV, visible and IR region, that is your infrared region, is caused by a recombination of electron hole pairs in the object, which is also called as cathodoluminescence. The basic construction of an electron microscope consists of these standard components: an electron gun, electromagnetic lenses. There are three types of electromagnetic lenses: condensing lens, objective lens, projector lens, and the whole apparatus is situated in a vacuum pump. This is a diagram of the basic construction of an electron microscope. The electron gun is a heated tungsten filament. There are basically three types of electron microscopes: transmission electron microscope, scanning transmission electron microscope. The applications of electron microscope, it is used in the study of biological specimens, in the study of macromolecules, metals, crystalline materials, surface characteristics of metals, organisms, and also in the inspection, quality assurance and failure analysis of semiconductor devices. This is an image of a mite under electron microscopy. These are the references. Thank you.